Hello, everybody. Thank you for giving me the opportunity to speak in front of you today. So my name is Brizzee. I am the Director of Gene Editing at Demeetra. And a little bit about Demeetra. Demeetra is a gene editing company located in Lexington, Kentucky. And we were founded in 2019. And about 80% of us do hands-on research. So we're PhD scientists, and we're really focused on developing and optimizing our gene editing technologies for commercial applications. And being hands-on research, we're able to really transfer that know-how and those protocols to our clients and work really closely with our clients almost as a collaboration or partnership. We have a really great innovative licensing and commercial product business models for agriculture, bioprocessing, and cell line development. Now, as you heard before, an issue is having that consistency and efficiency of the gene of interest or the antibody that you're trying to express. Random integration often results in cell pool heterogeneity. This leads to extensive screening as well as genomic instability. An option to solve this is through targeted integration. This enables consistent clones to be made, and it significantly reduces that time for cell line development. And this can also become very, very important when you're trying to put in these complex molecules, and we've seen plenty of talks earlier about doing these bispecific and trispecific antibodies, and really having a lot different expressions for these complex molecules. You want to be as consistent as possible. Now, one way to solve this is through an integrase-mediated integration. However, these integration techniques or strategies require very extensive landing pad construction. So first you have to put the landing pad in, which is a problem as well. But when you have these landing pads in the genome, your integration of your integrase or recombinase is often still pretty low. Now, there's an example with 5C31. Even with the landing pad, it's only about 13% that you're getting your GFP construct in. A lot of people use the BXB1 serine integrase. It's one of the top integrases based off of accuracy and efficiency. However, there's a lot of engineering that has gone into making it better, and still you're seeing about 7% efficiency, and I think the top number in this publication was around 20%. Everyone's used Cas9 to do this as well. This is one option to knock in constructs. So CRISPR-Cas9 is a monomeric protein. It has a blunt cutting mechanism, and it often leads to small indels, so you see about 1 to 6 base pair deletions. However, you do have frequent off-target events. And you also see very low targeted knock-in efficiencies. One of Demeetra's main core technologies is called Cas-CLOVER. This is a high-efficient targeted dimeric nuclease. This uses an inactivated Cas9 protein, and then it's fused to our proprietary CLO51 endonuclease. And this is an obligate homodimer, the CLO51 molecule, so it needs its partner to actually do the cutting. And this leads to very low off-target effects that you would typically see with CRISPR-Cas9. It has a really easy guide RNA plug-and-play infrastructure, so you could use the same PAM sites, the same 20 nucleotide base pair guides. And with this cutting, it leads to larger deletions because it has overhanging staggered cuts. So having a fusion protein that Cas-CLOVER is, fusion proteins are often very dynamic, and they're difficult to express and purify. Fortunately for Demeetra, we have produced this recombinant Cas-CLOVER protein that enable us to do in vitro assays to define its cutting mechanism. And here we have showing sequencing from digested products with Cas9 and Cas-CLOVER. You can see the left guide and the right guide that we use for Cas-CLOVER. And you can see the blunt cutting mechanism from Cas9, which is very typical right next to the PAM site. And you can see this 4-base overhang that Cas-CLOVER produces that's kind of right in the middle between those guides. And with that staggered cutting mechanism, we see higher knock-in efficiencies with Cas-CLOVER. Cas-CLOVER is proven in many different model organisms. We've used this many a times in tobacco. This is targeting the PDS gene, which creates an albino phenotype. We've also done this in yeast, targeting AD2, which creates a synthetic precursor that creates a red phenotype. We've also done E. coli, which we're hitting an essential gyrase A gene, and this creates a death phenotype, so you don't see any growth after the edit occurs, and as well as a lot of mammalian cell line work. And these are the typical deletions, if I can get this right. These are the typical large deletions that you're going to see with Cas-CLOVER, and they can be even larger. We also have improved indel efficiencies compared to Cas9, and this is in CHO cells targeting probably the most popular genes to target glutamine synthetase, as well as alpha-1,6 fucosyl-transferase, or as FUT8. With Cas-CLOVER, using many different guide RNA scaffolds, so this could be from IDT or Synthago or a two-part guide RNA system with IDT, you're seeing very consistent 75% cutting efficiencies, and this is with a T7 endonuclease assay, whereas in Cas9, you're seeing about 50% cutting. And in FUT8, you're seeing a very similar situation, so around 80% cutting efficiency at this certain exon, as well as about 60% with Cas9. So not only can we knock out genes very efficiently with Cas-CLOVER, we're seeing enhanced knock-in efficiencies with Cas-CLOVER as well. So this was a collaboration we have done. This was knocking in GFP with a puromycin selectable marker. This knock-in construct was about 7 kb. I wouldn't say that this is a huge knock-in construct, but these are relatively larger than you would kind of see. So we did this in HEC293 cells, and without selection, this is right after transfection, we're seeing around 10% of knock-in efficiencies. Compared to CRISPR-Cas9, you're only seeing about 2.5%. With a little bit of selection, about a week or so, you're seeing that Cas-CLOVER pool is enriched to around 70%, whereas your Cas9 pool is still about 35%. So higher efficiencies for Cas-CLOVER. One of our other core technologies is our HarborN license-free transposase. It has all the same benefits as Super Piggyback, but it falls outside the patent scope, and we exclusively license this to Lonza for mammalian cell work. But we do have our HarborN that is a license-free. It doesn't come with any IP restrictions, and there's no license required. We simply sell this as a commercial reagent. It's optimized mRNA that's going to give you copies around 3 to 5 copies in CHO cells. And our first-time customers with this, we provide a HarborN starter kit. So you're going to get the transposase in the mRNA form, and you're going to get a lot of transposons that can help you just go right in and plug your gene in. So this you can use in conjunction with a CHO cell that maybe has GS knocked out. It has the glutamine selection in the transposon, and it has multiple cloning sites, so you can play with all your different genes of interest and put them in. And this is available as a commercial reagent. You can use this for cell line development, bioprocessing, you name it. So using both of Demeetra's technologies, we worked with VBI Vaccines and the Coalition for Epidemic Preparedness Innovations, or CEPI, to create cell line platforms for a rapid response to pandemics. This was to create enveloped virus-like particles, and EVLPs are great because they structurally mimic enveloped viruses such as the coronavirus. And our first goal was to create a stable cell line that expressed the core GAG protein. So GAG is a structural component of these EVLPs. With increased levels of GAG, you will see higher EVLP production. And then secondly, we wanted to go in and create rapid and tailored expressions of these antigenic proteins. So these could be spike proteins with different epitopes at specific ratios that would lead to better efficacy in animal studies. One way we did this is we used the HarborN transposase to actually identify novel sites into the genome for a wide range of GAG expression. And then we went back in and actually did targeted knock-ins with Cas-CLOVER of these different VLP cargoes to elucidate which sites are giving or contributing to different expression profiles. So here on the top is our typical harbor in cargo. So these have your ITRs, your core insulators, and your protein of interest. After you do transfection, you can do rounds of selection and do single cell cloning. These are in HEC-293 suspension cells. And then we performed what we call a Splinkerette PCR. This is specific towards one ITR of the transposon, and then it's going to be able to identify the unknown genomic region where the integration actually happened, so you can actually start mapping out where your integrations came from. And we had multiple clones that we found that had around 4 integrations at different sites with different GAG expression profiles within ELISA. But we did notice that one clone down on the bottom here only had 1 integration, and it had very high GAG expression at chromosome 2. So we were really interested in actually exploring this site further because this could be a potential hotspot. So in order to work through and compare this novel site at chromosome 2, we went to the literature, and there was a paper by Aznarian et al. In 2022 that described different genomic safe harbor sites. And we chose Rogi1 and GSH31 as those were kind of a high producing safe harbor site and a medium expressing safe harbor site. So we wanted to compare the expressions of trying to knock in at all 3 of these spots. So we typically design 3 guide pairs per target region with different ratios of guides to your Cas-CLOVER. Now, we were able to find very good guides for each site, and this is important because your knock-in is only going to be as efficient as your guides. So if your guides don't work, you don't get any cutting and you don't get any knock-in. So we picked the best ones and then proceeded with the knock-in. This is essentially the same cargo that we're knocking in as the harbor in transposase, except now we don't have those ITRs or core insulators. We actually have 500 base pair homology arms, 1 knock-in construct with 6 kb, and then we also increased this to 12 kb, so this actually had GAG protein plus a spike protein. So we wanted to kind of complex them and put them in. So throughout selection, we're continuously monitoring these cell pools. And as the cell pool is enriched for the knock-ins, you're able to identify the PCR becomes a little bit more pronounced. So you're seeing better bands. And then when you sequence those bands, you can identify that you do have a knock-in for your clones. We found that for chromosome 2, it had a really high GAG expression that was comparable to GSH31 safe harbor site. However, Rogi1, that was supposed to be the highest, actually showed less GAG expression. And this is because these are enveloped virus-like particles, so they're actually getting exported out and secreted from the cell. And if you have too high of GAG expression, you actually start messing with the cell membrane and the cells are starting to die. So that's what we found there, but it was really great to see that you could use both these technologies to really identify these novel hotspots in HEC-293 cells. Another option that we have, we're getting in for cell line development, is a commercial clean-cut GS CHO cell platform. This was produced by our sister company, Harrow Biolabs. They're kind of right next door to us, so we work very well with them. Glutamine synthetase is still the industry benchmark for rapid CHO cell clones with high titers. So we have really good editing efficiency at glutamine synthetase. We have clones that have these large deletions, and then we have a great phenotype with glutamine behaving just like wild type CHOK1 cells, suspension cells. Without glutamine, you're seeing death within about 9 days. With these clean-cut GS CHO cells, again, this is some data from our sister company, Hera. They're using this in conjunction with the Harbor N transposase, where they're expressing trastuzumab, and they were able to get very good clones, stable trastuzumab expressing clones with Harbor N. And in unoptimized 24 deep well plates, fed batch, you're seeing cell-specific productivity around 110, and you're seeing antibody titers around 5.5. We took this farther with another CDMO collaborator, and they were able to take this into the Amber 250 and get about 8.5 grams per liter. So really nice cell line platform, and this is more than triple of the cell-specific productivity that I've seen with other transposase technologies such as leap-in. Something that Demeetra is continuously doing is we're working with Hera and we're trying to get different cell line platforms for clients. We're creating a clean-cut CHO, GS, and FUT8 knockout, so this is going to be a double knockout cell. We've already done initially really great gene editing. We've identified clones that are very consistent with 100% indels. There's some clones that have about one allele edited, and through a qPCR for gene expression, you can see that these clones have quite the knockdown of FUT8, or 6 through 22 is basically no FUT8 gene expression. Going a little bit step further to characterize these cells, this is the natural, clean-cut GS cells that have no editing at FUT8. You see nice protein staining of the FUT8 protein in green, whereas our top clone, you see no FUT8 expression. And then for a LCA staining, which is staining those fucosylated N glycans, you can see nice surface staining in our GS clean-cut cells, but our top clones, you are not seeing any of this fucosylation. We really wanted to take it a step further and try to push the boundaries of how big can we knock in a construct with Cas-CLOVER. And I know there's been a lot of talk about, it's really about the design and how to put in knock-ins. This was a very kind of simple proof of concept where our first one was 7.6 kb with 1 kb homology arms. We had 2 different proteins with glutamine selection, as well as COP-GFP as kind of being a reporter. And then we took this farther of putting multiple copies of these genes in and said, if we can knock in one copy of this with multiple copies in that knock-in, we're gonna see, hopefully we'd see greater titers right from the get-go. So we made knock-in cassettes at 7.6 all the way to 25.6. So that was fun to make, but you could, it wasn't too difficult. 72 hours post-transfection, we actually saw around 25% of positive cells with that 7.6 kb knock-in. What was even better was we saw pretty good knock-in efficiencies with the 21 kb and the 25.6, around 5%. And this, with Cas9, the theoretical maximum is around 14 kb I've seen. So we replicated this with Cas9 as well, and the cells didn't survive 72 hours post-transfection. Now we can take these and do glutamine selection, and you can see that this knock-in had really nice viability throughout the time frame, and you were able to enhance that population quite well, whereas your plasmid only was dying probably, it was pretty much dead before day 20. This was a slow glutamine selection. You can always modify this to enhance it or speed this up. One way to do this is you could cell sort. We have that GFP reporter. So we have these cells right now, and in the future we're gonna look at validating the knock-ins at the correct spot, as well as looking at gene expression and protein expression of these knock-ins. So just as a little summary, Cas-CLOVER, very low off targets since you need that 2-guide system. The knock-in's pretty is enhanced over Cas9, where the knock-in or the knockout is very comparable. Your indel sizes are a lot larger, so it's easy to characterize. Even with 2 guides that you need for this system, we're still able to frequently find guides for each target. The best part about Cas-CLOVER is we have really good commercial freedom to operate with a very simple license over Cas9's kind of complex, burdensome licenses. This is our Harbor In compared to Leap In. We're seeing that this is outperforming Leap in a variety of ways, but we provide this as a commercial reagent. It's in mRNA. We have some protein, but we're seeing less copy numbers with the protein. I'm assuming that that's because the protein's not in there for as long as the mRNA is. So it can be utilized for cell and gene therapy if need be. And we're seeing way more cell-specific productivity. Here was like around 25 copy numbers for Leap In, but you're still at a very low cell-specific productivity. Of course, this could be very molecule dependent as well. Lastly, I just want to conclude that Demeetra has a very comprehensive freedom to operate cell line development package. We have a really great license with the CVC. It covers the single guide RNAs, the 2-part guide RNAs, as well as the RUVC and the H&H mutations to inactivate Cas9. Helmholtz-Munich has the IP behind the proprietary CLO51 endonuclease, where our sister company, Poseida Therapeutics, owns the Cas-CLOVER system, where we have the exclusive license to use this in bioprocessing and cell line development, as well as agriculture. And we also have been making improvements on the CLO51 endonuclease as well. We also have we developed the clean-cut GS-CHO cell line with Cas-CLOVER, so automatically we're providing this cell line as a very kind of cheaper option with a simple license, and we also have a license through ECAC as well. And the Harbor In transposase is seeing very high titers and cell-specific productivity, and we're offering this as a commercial product. If you have any more questions, I'm available. You can message me on the app, or I can see you during the networking events, and as well as my colleague Koi Safan. She's our business development manager, and she will be around to take meetings too. And with that, I'll take any questions.
Very interesting talk. I'm wondering that if you have the Harbor In and then you knock in basically a bunch of constructs and then you select, and then also on the other hand you have your Cas-CLOVER. Have you tried them side by side to see which one of them has higher titers or would be more efficient for cell line development?
Yeah, so we're in the process of that. We really wanted to use Harbor In conjunction with Cas-CLOVER, so we would have stable integrations at highly expressed sites, and then if we have known spots that we want to target knock-in to increase titers, we want to do that as well. So we actually want to combine both of them and create a very high titer cell line.
So basically you have some that goes targeted and some that goes by the semi-targeted.
Yeah. So Harbor In semi-targeted. So piggyback transposase is notorious for finding those highly transcribed regions in open chromatin. And then if you can find those hotspots, or you have known hotspots, you can knock in with Cas-CLOVER and continue to use and put more knock-ins in. So with that, the HEC 293 project with EVLPs, we have all those different integration sites from Harbor In, and we can individually knock in different cargoes with different selections at those sites.
And one last question, any concern if you use them in the combination, the GS that goes into the Cas-CLOVER would provide enough resistance for your cells to be basically not needing glutamine so that the other ones don't have as many copies for the semi?
Yeah, so that's always a problem. I think we've played with the notion of really engineering the knock-in construct. So we have really a low promoter for the GS or glutamine gene. So you can get like more copies and you would still have enough glutamine getting produced where it's not going to mess with the other sites. If you have a stronger promoter, I think you might affect it.
All right, thank you.
Thank you for the talk. John Herberger from Melon Pharmaceuticals. It's a question on the, sorry, I'm over here.
I don't know where, I'm just seeing a bright, okay.
On the licensing agreement, I saw 4 companies on the last couple slides there. So who do you actually license with?
So we license with small companies or big companies, whether you're in the process of developing.
Sorry, but the company would be licensed through Demeetra?
Yes.
Exclusive, okay.
Yeah. Yep.
The other companies that are involved, they're underneath that license agreement?
Oh, so that is strictly for Demeetra. So if we license to you,
Yeah.
that's the CVC stuff is something that Demeetra takes care of and pays. We pay a small fee. We have a really great license with the CVC. That's something that they don't typically do. But yeah, you would just deal with Demeetra. You wouldn't have to get the sub license stuff.
Right.
Yeah.
Okay. Super. Thank you.